evolution you'd like to do in the lab. The big question is here, can the lab, being the early Earth, make this from scratch? Can you get evolutionary, first physical selections and then functional selections? Is there something in there which you can reconstruct and try to rebuild in the lab? And that's one of the starting points we did a while ago, where we basically took a protein. Of course, we cheated here. The protein was replicating the DNA in a temperature difference. These are small, tiny pores, where you see here the convection flow of the liquid between warm and cold. And there's an interesting effect that as molecules circulate between warm and cold here, up and down, hope you can see my cursor, they actually accumulate by an effect of thermophoresis.
We can see your cursor.
Okay, perfect. So that molecules have the tendency to go to cold, and they get shuffled in this flow, and therefore you get this accumulation. And the combination of it was actually quite nice, already getting close to Darwinian machinery, because we feed in here continuously our small single nucleotides, which the protein uses to replicate as a normal PCR reaction. And then the strands are separating on the warm side, get recycled again, you get exponential replication. And this flow and this thermophoresis in combination has an effect that the longer strands are actually surviving in here. And you can look for a gel experiment here where you flush in different lengths of DNA, and you actually figure out, depending on the speed, how fast you flush in things, that the longer strands, initially all the strands are inside, you crank up the velocity, and then you get quite a strong dependence on that speed, whether the longer strands are actually staying inside. So in a way, it can trigger length selection and replication. So in that sense, it would be an interesting setting to think about this physical emergence of replication, but we had to use the trick of the polymerase, which of course doesn't make sense, because polymerases need whatever 3,000 bases to end the ribosome you just heard about, and of course that doesn't solve the chicken and egg problem. So one of the big questions in the field is how you can do replication without having that nice polymerase. So what can be done before proteins? So we try to get one version of the origin of life scenario into these temperature difference chambers. This is a RNA protein ribozyme, so it's about 200 bases. It's collaboration with Jerry Joyce, just came out last summer. And here we have a chamber where we heat in the center, trigger a convection flow, and can actually tune it to the point that we have only short temperature spikes, and let the ribosome, that piece of RNA, replicate on a template. Again, it's a bit similar like a PCR. You have a template, you have a primer binding to it, and then you get the addition of bases to that. And it is short enough that the temperature spikes when it comes in here are actually separating the strands. And it has actually shown an interesting effect that these ribozymes have aggregated under these quite special buffer conditions, which Jerry Joyce's lab have optimized. And you can actually accumulate the ribozyme here in this area where it's a little bit colder. So it's an interesting effect here that by that aggregation, the molecules is saving itself into the colder side of the chamber. And therefore, because hydrolysis under these high magnesium conditions where these ribozymes are operating can be minimized, can be reduced. The interesting point is still to say that this ribozyme is quite a long piece of RNA, and it's of course not yet solving again the chicken and egg problem. How could you get to that molecule to actually replicate it? Even the second part we couldn't yet, while it's interesting to see that thermal oscillations by themselves can replicate things, we couldn't yet push the system to make replication of longer strands. So the mutations are so high in the system that basically the strand is just replicating so that the reverse primer from the other side, complementary side, to amplify back, similar to PCR, can't replicate any sequences in between. If you start with some sequences in between, it immediately mutates back to that minimal part. So we have not yet, therefore, managed to get this to a mode where you would replicate longer strands better. And that's because it's very heat selective. We can't use the geometry I just showed you before. So that's where we are. But, you know, as always, we have some ideas how to continue, and perhaps we get closer to really being able to replicate the whole thing in the Eppendorf tube. You know, in the tube, you can show that already, that you can replicate once, but then you have a double-stranded strand of RNA, which is really hard to separate, and that's where the field stands. Could we, you know, get over that strand separation problem? One way people are thinking about this is to make longer pieces like the ribozyme out of shorter strands and put them together by ligation. Okay, there are also ribozymes being able to do that, and that some Y ribozyme is actually quite interesting candidate to get to that. But of course, it would be most interesting if a chemistry could give you that ligation. Then you would, again, you know, heat things, strand would separate, cool it, ligation happens here by chemistry, heat it again, cool again, and so on and so forth. So we thought we could go by a standard activation agent of EDC, which is quite a strong, aggressive molecule to make this reaction going. And yes, you can actually make the reaction work out to some degree. So things are replicating. If you do here a serial dilution of your template, you can actually replicate off that template. But this reaction suffers from the aggressiveness of that EDC. And you can look here for the details if you're interested in, you know, this is what our reaction could do if we optimized it. But basically, this is what we would have expected. We would have expected much more production of the template, but the EDC made a lot of additions and changes of the bases nearby, and just destroyed the reaction because it was too aggressive, also modifying the strands themselves. But that's the motivation, you know, can we go for the ligation, and would that ligation reaction be a way to get to a mode of replication more interesting for the first steps of living systems. And here we look into a system where we, on purpose, limited ourselves on the complete other side than I told you before. Before I told you, you know, we take an aggressive chemistry of EDC, we get fully covalent bonds. Here we kind of try to get away without any chemistry. Could we go for a replication system with all the know-how and the expertise of DNA assembly, hairpin formation, all those DNA nanomachines, by running a replicator just on temperature? The second motivation for this, we would start with a molecule which is known to be early, of tRNA. You know, can we design a replicator based on tRNA to do this? We'll get to the end what motivates us to do that. You know, it will not get to any synthetic ribosome, would be really interesting, but perhaps a little bit, some steps into that direction. So that just came out in eLife here on the right-hand side. So the starting material of how we activate those materials would be actually two hairpins, which are connected with a here colored part of the sequence, which puts up a binary code. These are 8 bases, 7 bases. It's either red or blue. And the idea of the whole thing is, as we heat it to 90 degrees, everything would separate into small pieces. If you cool it down, things would first make those hairpins, and those hairpins will close up the interaction with it so that they don't bind to each other. So if we go for a first step and go for 45 degrees, where, you know, in principle, we could think about that these hairpins could assemble and come together. They don't, because they've been quenched in this metastable activated state of having the hairpins. If in that process of cooling, we actually offer it the template, and the template is made out of the same molecules, which if they come close, if you heat them up, if you make a very slow cooling down, for example, they are building these connections. And that's kind of the analog of what the EDC molecule was doing in the ligation of putting the two strands together, because those give a long stretch of double-stranded strand here, and therefore, not melt up so easily. So if that template is in here, we give these hairpins, the hairpins can bind to these color-coded regions in this sequence 001, bind there, and because they are then in proximity, the high local concentration and the fluctuations of the hairpins will allow us at these temperatures to fully make the complementary part. You see the logic. If we take the reverse complement on the other side, these double hairpins come from here, bind here, and also get to the same construct. Now, the interesting point, or that's how we try to deal with this, is if we are only heating now to 70 degrees, it only melts here between those informational domains. And since we still have the hairpins, they can come in and actually replicate. So if we cycle this between 17 and 45 degrees, that's the idea, you would get an exponential replication of that signal out of it. So in a way, you can think of as if a 3-level laser. You have an excitation at high temperature of pumping the system to create these small double hairpins, go to the upper laser level, 70 degrees, and then you get stimulated emission. So if you have a template, you get off the template and replicate the template. So again, coming back to that muddy pond in the first slide, how would you think about it? It would be that you have outside convection flow, which goes for full high temperature of 90 degrees. Those will create these double hairpins. And inside, you'll have other laminar flow chambers, which would oscillate nicely between this about 70 degrees and replicate from there. And basically the outside would, by diffusion, feed into the flow into the inside and continuously feed these activated molecules. That would be the idea. And the experiment trying to track this system has been done on native page gels, trying to see for these constructs, I think actually on the friction coefficient to really understand the system and optimizing it, right? So on the first hand side, we look out what's a very good temperature for this assembly, whether it's coming here. So 45 degrees is a good compromise where we are not yet getting false positives if we don't have any template inside. If we look for the concentration dependence of that assembly with the template, it shows a reasonable concentration dependence as expected if template is triggering the assembly. And then you can choose the upper temperature, about the 70 degrees, where you can actually separate the strands and run a replication reaction on it. Now the full question comes about, so how much is this growth of these templates concentration dependent? Gives a linear dependence, as you would like to have for cross-catalysis. If you start with one side of the template, you get these curves which show already a little bit exponential growth because you start only from one side, it's not yet starting immediately. And the major test is to run for a zero dilution experiment. So basically you let it replicate for 3 cycles, dilute by a factor of 2, let it again replicate from what's left, and so on and so forth. And the system survives reasonably well if we give them all the food, and actually goes down onto the zero dilution if we don't give one side of the food. So it's not exponential. So that's a bit the story, what we can do if we only run things thermally. You can have a replication fidelity discussion, which is based on if we remove one of the foods, and yes, you get about 70% of replication fidelity. But what is interesting for us here is that we kind of start with a tRNA-like starting point and make a replication reaction out of it. So how would nature select such a machinery? Why would such a machinery, be selected from random sequences? One indication for that would be that if we make the system be capable to make long replicates, it can actually form aggregates. This is just gravity-induced sedimentation. You can look down here that over a course of a day, these molecules get so large that they make aggregates and they fall down and sediment. And we only see that if we have the full combination of all the hairpins you need to make that full construct. If you have either the blue ones with a 0 coding or the red ones with a 1 coding, you get the sedimentation. If only one of the 8 molecules are missing out, you get no sedimentation. That could be a way to select such a construction. And that works reasonably well if we omit any of these 8 strands. So the idea here would be that temperature is not only activating these molecules, making them ready for replication, but you could have also a pocket in your convection stream where they would sediment and save themselves for long-term storage. There's a lot of double-stranded parts here, so hydrolysis will be weak, and you could think that that will be a selection pressure for surviving such molecules. Still, it's again the same story as before. We ask for 70 bases and 8 molecules, 70 bases, which don't have to be defined on a single base level as for ribozymes, but still, it's a little bit a steep curve to ask for. But we're kind of fascinated by the fact that you can think about a very old molecule, and with 3 mutations, you can refold it into a double hairpin. And then if you take 7 other molecules, which are actually not tRNA molecules, but already designed and optimized to make this game, you could go for a replication scenario. So how much this, would be able to go and describe early generation of coding for amino acids and making proteins and peptides, that's up to your fantasy. It's a bit a wide step, but still you can think that this refolding brings this CCA end here of the tRNA close to the anticodon and could select amino acids here, fish it out of solution, bind it, and therefore encode here the color coding of blue and red would be accordingly encoded in amino acids. And you just see in the talk of Andrew how you can run that in a synthetic ribozyme. That could be something where schemes like that or similar ones, or it's a bit up to your fantasy how much you can think of that this could be a way to bridge from replication to translation of proteins. Okay, how am I in time? Okay, fair enough. So as you see here, we are fascinated by the ligation. Here the ligation is brought about this hairpin sequences. You've seen before we tried to do the ligation chemically. And here we kind of did away and just assumed if we take a protein to do the ligation, what sequences are we getting if we start from a random sequence library. So yes, again, we do the trick of using a protein, but a ligation reaction might be the most simple reaction you could think of to make. And the interesting story here is that we started with random sequences, make this ligation, and the only way we keep it out of equilibrium was doing a temperature oscillation. And that temperature oscillation was, interestingly enough to keep it in a quite interesting non-equilibrium setting, in a way that the outcoming longer sequences actually did not show at all random sequences anymore. So we actually found here a system which is reducing the entropy of the sequences even by a very simple process. So that just came out in PNAS, and these are the length-dependent distributions. So you have, initially you have basically mostly monomers, and then you actually create a long stranded of 180 mers in the process, and then we took those for Illumina sequencing and analyzed the sequences. And initially you might say, why should it ligate at all? I would expect that if you have 12 mers, give it enough time, and that's exactly the point, they should hybridize like this, and there should be no ligation at all, right? If you have a hybridization of this time scale and estimate with it, you can compare how long it takes to make that 12-mer random sequences hybridize, and these hybridizing, where you basically have 2 times the 6 mers, which are independently hybridizing. You actually figure out that this can be pretty fast if you take 2 bases of A and T, as we did in the experiment, 6 seconds versus 410 seconds. So if we do a temperature cycling, which is already cycling here back again to high temperatures, this will never settle significantly in that construction, but in this construction. Even to the point, if we try to make it with 4 bases, this would be 19 days versus 410 days, 410 seconds. And that we saw in experiment. If we tried to do the experiment with 4 bases, we actually never saw any ligation in our product because it just did not have enough time to get this construction, because the dilution of concentration in the sequence space was just too long. So that's an interesting interaction between temperature cycling and size of sequence space, which is pushing the system already for picking low amount of nucleotides for replication. Okay, so that's a protocol. We run between 25 degrees and 75 degrees. Run now with AT only bases, and now you can actually vary the ligation time and actually really probe for this. And so if things are shorter than 30 seconds, we are not getting any of the long strands. If we give it a little bit more time, 20 minutes or even 1 hour, it actually goes down. Also, because you can offer in the constant time you give it, about 66 hours, not enough cycles to ligate. So this is what to be expected by the arguments I just showed you before. So what do we get? So we let it run for 1,000 cycles, analyze the sequence, and we start with a 12-mer, which has a reasonable random distribution, gives you normal binomial distribution. And already the 24-mer of it actually gives you a bimodal distribution. So what this can be seen here is that you either have molecules which have a lot of A on the first part and a lot of A on the second part, which is completely correlated, or T and T. You never see here a lot of A and here a lot of T because, well, they just make hairpins and then can't participate anymore in the ligation and the templating of it. So the system, although it's very simple, is giving you selection for not forming hairpins, but it also gives you a quite interesting network of sequences which can help each other to template again the next round. It's a bit not enough time to go into the details, so this is actually accepted now. Sorry, that's the old slide. You can actually see that you have sequences which have a lot of A, which make networks of trying to template the reverse complement on the other hand side. So it really, the sequence space structures itself for templating. And you can understand that system by really creating networks where you are expecting to get a network, or you're expecting to get very fast elongating sequences and fully make use of that understanding. So that as we want to go for those ribozymes of the RNA world, templating looks quite interesting to give you a way to get from random sequences into highly structured sequences already at the stage where you just want to ligate things on the template. So that's a new insight in terms of how to reduce sequence entropy. If you want to do that, ligation would be a really interesting protocol. So better look for that. So that gives you a way of symmetry breaking the sequence space. And go for that. So just closing it up, I want to make a short remark that we are very interested in kind of microdistilleries, where we have, again, a porous rock sample where we have, and think about molecules and liquids in that temperature difference. But here now we have a pore which has a little bit air, CO2 also in between, and that triggers evaporation condensation cycles as you make the temperature difference. So things are accumulating here, and that accumulation gives us quite a strong, fast accumulation of nucleotides, oligonucleotides, of salts, and of RNA enzymes. So this is now RNA enzyme which is cutting a substrate, and you see that by fluorescence, and it's really active very much at these interfaces. So there's air on top, and at the bottom this is liquid to the point that it really fills the whole interface. And we could actually observe here a number of interesting effects which periodically would be interesting. So our ribozyme catalysis, chelation, so things are aggregating again into big aggregates, encapsulating DNA, RNA into vesicles, crystallization, and also phosphorylation. And we're starting to continue here to get also polymerization into the mix. So that's kind of a bit the update to get you a little bit an overview of what can be done, what can be pushed for understanding autonomous evolution. I'll skip over these slides here. This is kind of the machinery we do it. We have some microfluidics and Sapphire, and we have built and made a lot of collaborative links in Munich. We have Emergence cluster, which gets funded for 7 years now. Hopefully we'll get the extension. We have a CRC Emergence of Life. Again, this is 4-year chunks. We are writing right now the extension of this one, and I'm part of the science collaboration on the Emergence of Life, which really brings us to the next level of collaboration between all the disciplines. And with that, I want to thank my group, and happy to get questions in, what's that, room number 2, right?
Indeed. Thank you.